George, thanks so much for accepting our invitation and being with us virtually today. We would love to host you in person post-pandemic, hopefully sometime soon in the future. Thank you all for coming and joining us virtually together today for the 76th R.A. Mashelkar Endowment Lecture, a very illustrious series of talks sponsored in part by the NCLRF Foundation. It's indeed a great opportunity for all of us to get together to hear Professor George Church share and talk about his extensive work and incredible ideas. I have happened to hear him on more than one occasion, and his talks are definitely incredible and very inspiring. George is someone who almost does not need any introduction. He's someone who represents a leader and a visionary figure in the field of genomics, that is reading, writing, and revolution in DNA, and is practically the father of genome sequencing. It's an honor and pleasure to formally welcome and introduce George Church, and I would like to invite and request our director, Dr. Ashish Lele, to do so, who himself is a leading figure in polymer rheology, and more recently, he is a leading figure in fuel cell technology and hydrogen energy. Ashish, the stage is yours.
Thank you, Anu, and thank you, Professor George, for accepting our invitation. And as Anu said, we would love to have you here in person, visit us in India the next time it allows us. So thank you very much for your time. Professor George doesn't really need a long introduction, but as formality, I'm going to do that anyway. So Professor George is a professor of genetics at the Harvard Medical School, a founding member of the Wyss Institute of Biologically Inspired Design, and director of personalgenomes.org, the world's only open access information on human genomic, environmental, and trade data. He's known for pioneering the fields of personal genomics and synthetic biology. Professor George leads synthetic biology at the Wyss Institute, where he oversees the directed evolution of molecules, polymers, and whole genomes to create new tools with applications in regenerative medicine and bioproduction of chemicals. Among his recent work at the Wyss is the development of a technology for synthesizing whole genes and whole engineering of whole genomes far faster, more accurate, and less costly than current methods. He is widely recognized for his innovative contributions to genomic science and his many pioneering contributions to chemistry and biomedicine. Professor George developed the first methods for genome sequencing that resulted in the first genome sequence, the human pathogen H. pylori. He has been involved in developing methods for dramatic cost reduction since then, which has come down from $3 billion to only about $600, which is an incredible cost reduction, and contributing to nearly all the next generation sequencing methods and the companies. He helped initiate the Human Genome Project in 1984 and the Personal Genome Project in 2005. He invented the broadly applied concepts of molecular multiplexing and tags, homologous recombination methods, and array DNA synthesizers. His many innovations have been the basis for a number of companies, including Editas, Gen9 Bio, and Veritas Genetics. Professor George's team invented CRISPR for human stem cell genome editing and other synthetic biology technologies and applications, including new ways to create organs for transplantation, gene therapies for aging reversal, and gene drives to eliminate Lyme disease and malaria. More recently, the tech entrepreneur Ben Lamb is helping fund an ambitious and science fiction-like project by Professor George to revive the woolly mammoth, a creature that went extinct 4,000 years ago, and create a calf in as less as 6 years. And this is something that has caught the fancy of the media, and all of us have obviously been hearing this. Professor George is the director of IARPA and NIH Brain Projects and the NIH Health Center for Excellence in Genomic Science. He has co-authored more than 500 papers and 130 patent publications and one book, Regenesis. His honors include the Franklin Bauer Laureate for Achievement in Science, the Time 100, and election to the National Academies of Science and Engineering. Professor George strongly believes and advocates that the best way to predict the future is to change it. And so without further ado, let's hear what Professor George has to say today on analysis, synthesis, and imaging of large molecules. Over to you, Professor George.
Thank you very much. That's a very kind introduction, very generous. I will share my slides. Can you see and hear me fine?
Yes, we can.
Okay, so the title is Analysis, Synthesis, and Imaging of Very Large Molecules. These are biological macromolecules, DNA, RNA, and proteins, and all the things you can make from it. I have a conflict of interest list on a website in the lower right-hand corner, and I'm very indebted to these various organizations that have helped our work get into the public domain. So we cover a broad expanse of scales, probably from the yoctogram, meaning a single atom changing from a nitrogen to an oxygen between an A and an I in the base pairing of DNA. So this is an adenosine deaminase. I'll tell you more about that in a moment. All the way up to ecosystem scale considerations. It remains to be seen how big an impact we can have on this, but there's 1,400 gigatons of carbon that we're in the Arctic that we're working on making sure stays sequestered rather than being released as methane, and we add to that additional carbon dioxide in the form of cellulose. So that's the span from essentially one Dalton, to 1,400 gigatons. And part of the reason we can talk about this has already been alluded to in the kind introduction, which is that we have almost a 60 million-fold improvement in the cost and a similar improvement in the quality of both reading and writing DNA. And my lab has been a big component of both of those revolutions, and they continue. It's not clear what the upper limit is for the productivity of reading and writing DNA, but it's probably several orders of magnitude, several factors of 10 more than where it is now, and largely due to multiplexing, which is not parallelization or automation per se. It is the mixture of bar-coded molecules. So a long, normally expensive process is about the same cost for one category of molecules or a million or a billion. So you can mix them together if they're properly bar-coded and decode them at the end. And that's multiplex next-gen sequencing, multiplex chip synthesis of DNA, and various multiplex cellular assays. I'd like to celebrate the world's progress on gene therapies, ranging from the most expensive drug in history, which is Zolgensma, which is aimed at a rare disease, spinal muscular atrophy, or SMA, to 3 companies that have used variations on gene therapy to deliver vaccines. So AstraZeneca has an adenovirus vector, and Moderna and Pfizer have a lipid nanoparticle delivery, and they both include a single gene, either in DNA or RNA form. And this has brought the price down from $2.5 million for a rare disease to as little as $4 per dose for basically a gene therapy. And that's because of the size of the market. It is a big factor there. So when we talk about gene therapies and cell therapies, cell therapies often are ex vivo gene therapies. We have 4 basic categories of editing. This is broadly defined editing, meaning any kind of genome engineering, really. So you can add genes that are missing, you can subtract genes. Precise editing, which is more subtle than either of the first 2, and epigenetics, where you don't affect the DNA sequence, but you change the way the DNA is regulated. And so, so for addition, we have transgenics and cisgenics, cisgenics being within a genome, trans being between 2 different species. Messenger RNAs is another way of adding, as we did with the, as we've seen with the vaccines. Nucleases is a good way to subtract, or I call it genomic vandalism, and then deaminases and recombinases we'll talk about in a moment, and various ways of getting epigenetic changes. These are some of the companies that that have contributed to these revolutions that I've been involved in. Now, in order to do this, we have to be humble. We have to know that we can't design the perfect right from the beginning. And so we make large libraries, and we use machine learning to refine the components that work the best and make another set of libraries. So we can do rounds of machine learning and mega libraries, which we call ML. And we can do that for protein design, we can do it for genome design, and we can do it for epigenomics. And you'll see examples of each of those in just a moment. This slide is just on protein design, and we published 5 papers recently. This is not based on predicting of 3-dimensional structure. It is one can do this without even knowing 3-dimensional structure, much less predicting it. This is done by using large collections, millions of diverse protein sequences to prime the machine learning pump so that we can make new proteins that are wildly different, as you'll see in just a moment. So these 5 papers, the authors have spun off 3 companies, Dino Therapeutics, aimed at AAV viral capsid delivery of gene therapies, Manifold Bio, that allows testing of thousands of protein pharmaceuticals simultaneously for low cost, and Nablo, which is working on new enzymes. So I'm just going to illustrate one of those 3, which is the AAV, the adeno-associated virus, which Pierce and Eric published 2 papers in Science and Nature Biotech that describe how we apply the machine learning and mega libraries, the ML, to making millions of capsids to test for immune evasion, which is important so you can reuse or use new capsids, and tissue specificity. So you either want to give it broadly to multiple tissues, which requires engineering, or have it very specific. And so I'll show you some of the primary data on that. Here we've made those millions of viruses where we've changed every position along the capsid gene, this is just a little piece of that capsid gene, along the x-axis, and along the y-axis is every possible codon substitution, every amino acid substitution. And then we sample where the virus goes, where it homes in multiple tissues. Here's 6 of those tissues, and red means that it's better than wild type and blue means it's worse. And so we're looking for little red dots here in each of these different tissues for tissue tropism. And that's systematically going through one at a time every possible substitution. But what if we want to make multiple at once? And so we took a small segment of the capsid, 28 amino acids, and did mutagenize all 28 at once using the machine learning from the previous one. Now you can't make all possible combinations of 20 amino acids at all 28 positions. That would be 20 to the 28th power. But you can use machine learning to guide this. And you can see if we do it randomly, either poor algorithm or random mutagenesis, we can only make 4 changes before we break the capsid. It doesn't work. But with machine learning and the large libraries and logistic regression in particular, we can get essentially substitutions at every position, every amino acid position with the right combinations. So this is a huge difference between random and machine learning. Now we'd like to apply that that we use for proteins and viruses to epigenetics of the cells of our body. We want to be able to generate every cell type from our body. And to do that, our starting point is a large international effort to read out the epigenetic state, the RNA state of every cell throughout human development. And this is sometimes called the human cell landscape or the human cell atlas. And that's just a starting point for us though. And it's not complete, but it's getting pretty close. We don't even know how many cell types there are, partly because there's fuzzy boundaries between the cell types in these 2-dimensional or multi-dimensional plots, where you categorize them based on their RNA transcriptional profile. So here's one from Han et al. In Nature. Now we've turned those observations into recipes, and we do that by using a transcription factor expression library. So we have expressed we think every transcription factor in the human genome, at least one expression clone for every open reading frame that could plausibly be involved in transcription regulation. This turns out to be over 1,700 such transcription factors, or TFs, and we have used these in various combinations to get up to 290 cell type recipes so far. And I'm just illustrating 3 of those recipes here. These are to make endothelial cells, neurons, and fibroblasts. And you can see here this heat map showing different transcription characteristic targeted transcriptions, RNAs, that respond to these transcription factors and produce morphology and behavior physiology of these respective cells, endothelial, neuron, and fibroblasts. Now, this is work of Alex and Peristu, who were postdoctoral fellows in the lab and now have started their own company, GC Therapeutics. Here's an example of experiment that involves 3 cell types, 2 of them shown, where the axons, or A, of the neurons are wrapped with myelin, M, from the oligodendrocytes. And this wrapping seems to be perfect, both by electron microscopy here and by function, as you'll see in the next slide. So this wrapping of the myelin sheath is what happens normally in our white matter in our brain and the spinal column to allow action potentials from the axons to go quickly through those regions. And these are the parts that get demyelinated in serious demyelinating diseases like multiple sclerosis. So we wanted to be able to treat those diseases by implanting this mixture of these 2 cell types and endothelial cells to carry blood to the human organoids. So the human organoids are implanted into the mouse brain in a mouse that has demyelinating disease, very analogous to multiple sclerosis. And we can rescue that disease by this sort of coordination assay. And this is the sort of work that we're doing in preclinical animal trials to hopefully lead soon to human clinical trials. Again, this is Alex and Peristu at GC Therapeutics. Now, that was an example of a transplant from humans into mice. But if you go to transplants within humans, normally that's challenging to do even with 2 human beings, not between species. And ever since 1954, when the first successful transplant was done, there's been a need to use humans that are matched at the MHC, the major histocompatibility, or HLA. And this is despite revolution in immunosuppression from the 1980s. We need both immunosuppression and matching of antigens. But that's changed recently, and there's been enthusiasm for the possibility of transplanting not just human to human, but from animal to human, a pig in particular, because of the proper size of the organs. And this happened as early as 1963, where 13 chimpanzee transplants were done, of which 1 worked very well. The human recipient lived 9 months, and on autopsy, there is no evidence of rejection of the chimpanzee kidneys. So fast forward from 1963 to recently, when 3 of the pioneers of the field, shown here, Martine, Joe, and David, invited us, Luhan Yang and myself, as pioneers in CRISPR, to join them on xenotransplantation. And one of the other inspiration for our work that I'll describe in just a moment was this idea of universal CAR T cells. So the CAR Ts attack B-cell tumors, leukemia, and lymphoma based on a shared antigen, CD19. They attack with addition of a chimeric antigen receptor. But the universal part is the key thing for this discussion, is that we want to make them so that you can take any donor and go to any recipient without HLA matching. And that universality is achieved by suppressing the major histocompatibility via beta-2 microglobulin deletions, this editing to eliminate certain factors like the T cell receptor, beta-2, PD-1, and various factors that make the cells susceptible to chemotherapy that might be going on at the same time. Anywhere from 1 to 5 of these knockouts of these genes can be achieved by various nucleases like zinc finger nucleases, talon nucleases, and CRISPR nucleases. And this is one of the first successful treatments with the CAR T in this little girl. And but that's 1 or 2 or 3 edits. We found that when we collected the wish list of everybody in the xenotransplantation field, it looked like more like 42 edits. And we've now finished all 42 of those edits and published them in these series of papers in Science and Nature of Biomedical Engineering. Luhan Yang was one of the pioneers that invented CRISPR in our laboratory. And she also co-founded these 2 companies that have taken these and similar germline edits of pigs into preclinical primate, non-human primate trials at 3 hospitals, including Massachusetts General Hospital, where Jim Markman is the surgeon leading the surgical team and testing these, and they look very promising so far. Now, the goal of that is not just to alleviate the worldwide crisis in inadequate availability of organs, but to make organs that are better. So some of the reasons that a person needs a donated organ doesn't go away when they get the fresh organ. So it could be infectious disease, it could be other environmental factors. And so just like the transplant from human to mouse that I showed earlier for multiple sclerosis like demyelinating was an enhanced set of cells that are resistant to the autoimmune response. So that was already the case for that one. And similarly, for some of the organs we're developing, we can make them pathogen resistance, senescence resistant, cancer, and even resistant to freezing and DNA damage. These have all been demonstrated in animals, and we're moving forward on introducing them into the pig organs. Now, in order to do that many edits, the 42 edits, or even 25 edits, which was our preliminary knockout of the endogenous retroviruses, we need to have we get toxicity. The double-strand break that comes from editing with CRISPR is toxic. And it's so much so that when we're trying to do 25 edits, we find essentially no single-cell clones that are appropriately edited. Well, if we use 2 empirically determined factors, a basic fibroblast growth factor and a p53 inhibitor, now we get almost everything as the correct correct edit. Set of edits, 25 at once, and you can do either all at once or you can do them in multiple rounds. And so in the end, we were able to make 3 edits to 3 different sugars that are different between animals and humans. The various immunological components, the clotting cascade, the complement cascade in the blood, and these endogenous retroviruses. Now, the endogenous retroviruses are a kind of repetitive element. It's been shown that the endogenous retroviruses from pigs are actively produced by every cell in every pig, and they will infect humans and replicate there. And then that would be a potential zoonotic disease that could evolve in the immune suppressed patient. So we really don't want that. The FDA didn't want that, so we knocked out all of their ability to replicate, leaving behind their envelope gene for various reasons. Now that's a repetitive family, and it was easy enough that it encouraged us to try other repetitive elements. Now UCEs are not repetitive in the DNA sequence sense, but there are almost 1,000 of them spread throughout mammalian genomes, each mammalian genome having the same set, almost perfectly conserved. Telomeres at the tips of chromosomes, ribosomal DNA repeats on 5 of our chromosomes, simple sequence repeats, centromeres that help segregate our chromosomes, lines and signs, which are about 20-some thousand and 1 million of them interspersed throughout the genome, and triplex. And these are not just interesting because they're unknown dark matter, but because they're involved in various health and disease components, including senescence, neurogenesis, cancer, and inflammation. And this is just a partial list of literature outside of our lab. Now, to edit them, like I said, the CRISPR double-strand breaks are toxic, so we had to do something more subtle, which is the deamination I mentioned in my very first slide, removing a nitrogen and replacing it with oxygen. The C, when you do that nanosurgery, you get a T or a U intermediate, so C to T, and A to G has an I intermediate, inosine. This can be done by either Cas9 or Tau fusion to deaminases. Luhan Yang, who I already mentioned, pioneered the Tau version, and David Liu's lab has been very generous and amazing development of both C to T and A to G deaminases, and has shared them with our group very generously prior to publication, including one which is CRISPR-free, similar to Luhan's, but specifically for mitochondrial editing. So we prefer the A to G because it has less off-target and it has less nicks due to repair processes. And so we and you can see that these have a off-target problem. Both the C deaminase has off-target both distant from the target, meaning where the C deaminase domain of the fusion protein has its own mind. It goes and it finds its own targets all over the genome, but it also has off-target product right near the target. So if you want to get one of these Cs, you end up getting all 3 Ts in this example, which we use for line elements. So we have to use all these different methods for making sure we fight the toxic effects of double-strand breaks and even little nicks due to repair. And this was published recently by Corey, Oscar, Khalid, Farina, and Raphael. And here we did it in human stem cells. These are pluripotent stem cells derived from the Personal Genome Project, actually from my arm. And we have a record of 22,000 edits per cell. So that's all in the same cell, all in a single transfection. And here's data showing, documenting a clone of a base editor targeted mutations in the line element. Now that's fine if you're aiming at repetitive elements, and sometimes if you want to ask questions about repetitive elements, you might need to alter every member of the family, and but if you want to do unique sequences, and you want to do a lot of them, as we want to when we're doing recoding, we're changing the genetic code from the 64 triplet codons, one of them to another, a synonymous one. So for example, TAG is synonymous to TAA in that they both are stop codons. We are in the process of changing the human genome so that all the TAGs at the ends of open reading frames, proteins, are changed to TAAs. And that gives us a number of uses that we'll talk about in a moment. But anyway, Yuting Chen and his team, both when he was a postdoc in my lab and now his own lab in Shenzhen, China, have seen how many unique edits we can do at once. And right now it's quite modest compared to the 22,000 that we can do in repetitive sequence. But we think this is going up quickly as many of the technologies are going up exponentially. We're at 33 genes, and not just genes, but essential genes that we've done in a single transfection. So we're putting in 33 guide RNAs, actually more than that, but 33 that work well. And we can do them in 2 ways. We can either put them in as a pool of DNAs that encode the guide RNAs, or a pool of RNAs without the DNAs, or a pool of DNAs concatenated into a large circular vector. This artificial chromosome can be introduced, and that has the advantage over the random pools in that the stoichiometry is fixed, and the regulation might be a little bit better. But we're doing both, and we haven't decided which one is better. But we want to move this up from 33 to thousands. We do this. We can analyze them by clonal expansion from one cell to many, and by FACS analysis. And we have software that keeps track of all the essential genes, all the essential UAGs at the ends of those genes, and how to edit them, what base editors are appropriate and efficient. Now we de-risked this. We played this out all the way to the end for one organism, an industrial microorganism, and one codon, one of the 64 triplets. And we did that every position in the genome, all 321 positions in the genome. And we're aiming for 3 new phenotypes. This is not just a structural effort. This is changing the 3 properties. So we can now efficiently incorporate non-standard amino acids. So whole new chemistries for the proteins, because we use this new codon. We can also do biocontainment. Using this new amino acid, we can make the organism addicted to it, an amino acid is not found in the wild. And finally, we can use the new code to prevent not just one, but many, or even possibly all viruses. We think that this method does work in one organism for multiple viruses, and we think we can make it work in any organism for all viruses. And so these are some of the papers starting in 2009 to 2020. Nili Ostrov was a recent leader of this project. She now has a large FRO, a focused research organization aimed at this kind of problem. And here's a summary of the codon table. Each of the numbers here are the number of codons used in this organism in each of the 64 codons, how many times it's used. And by far the most infrequent one, the rarest one, is this UAG that we changed to UAA. So all 321 of them were changed. So that in red box was the first one we did, success completely. Jason Chen's group added another 2 to that, making a total of 3, and showed that that also was multivirus resistant. And then we're almost done adding 6 more to that list. So 9 on the list, 7 at once. And again, that's work from Nili Ostrov. But the new leaders of the project are Akos and Anush, who hope to finish that project very soon. Here's data that these are truly multivirus resistant. We start with a input of plaque forming units, 1 trillion plaque forming units, trillion viral particles that are active, and we end up with 0 for all 5 of these viral categories. These are whole categories of virus. Doesn't matter whether it's single-stranded DNA, single or double-stranded RNA, membrane, capsid, or whatever. All they have in common is they need the host ribosomal codon recognition mechanism. The only 2 that escape are viruses that don't happen to use the UAG triplet codon in an essential viral gene. And we're fixing that by extending this to other codons, not just the 3 to 7 codons. This is work from Farron Isaac's lab. He was a postdoctoral fellow in my lab, but also and now is tenured faculty member at Yale and a collaborator still. Now, all the work that I have described so far on human cells has been done in, most of it has been done in Personal Genome Project cells. This is a project available to you. It is worldwide, and it's fully open access. Both the data and the cells are freely available in a nonprofit fashion, and they've been used by the NIH for the ENCODE project. Have been used by the National Institute of Standards and Technology and the Food and Drug Administration for establishing genome standards, and they're used for testing personalized medicine, imaging. This is a brain image from one of the volunteers. And this has been approved, the ethics has been approved in United States, Canada, UK, Austria, and Korea, and we hope to have one in India soon too. A lot of the sequencing progress that we've had, not just has been nanopore sequencing. They now have the longest DNA sequence reads, and you can see they're very small as well. This is this little box here is a nanopore. We hope that they will be part of your cell phone soon for analyzing not just things as exotic as the International Space Station, but your everyday exposure to environmental pathogens, allergens, and so on. But another large revolution in DNA sequencing is fluorescent next-gen sequencing. So these are not portable sequencing devices. They're quite large, but they have some interesting possibilities I'll mention you've already seen this slide on exponential improvement. So like I say, these are large sort of person-sized devices from MGI and Illumina. But they all, I would argue, are a form of microscopy, fluorescent microscopy that we started in 2003. And here's one that's based on the Nikon instrument that Jay Shinduri and colleagues published in Science. But they're a very limited kind of fluorescent microscopy in that they can only do a monolayer of molecules. They can only do a nanometer thick. And so that makes it hard for us to image a three-dimensional arrangement of molecules as occurs in cells or organs. So to get that three-dimensionality, we adapted the same kind of technology, but now to three-dimensional imaging as occurs in confocal, in biplane, and light sheet optics. And here's two different systems here. The ReadCore system recently acquired by 10x Genomics, and the Acuity Vutara Bruker family of instruments. And these are all three-dimensional. They're 1,000 to 10,000 times thicker than the conventional sequencing methods, and they handle DNA, RNA, and protein. And I'll show you an example of each of those kind of in closing here. This is in situ. These are typically fixed specimens, but I'll show you how we can also get time data that you normally would think of as happening in vivo. Now, these molecules, so these are the largest molecules in nature that I know of. They're of a defined sequence, and these are the chromosomes. And the largest human chromosome is about 250 million base pairs long. These are the same segment from two copies of chromosome 19. So it's a little piece of chromosome 19, this little red blob. And the reason there are two of them in the same nucleus, the same cell, is that you get one from your mother and one from your father's. So, and you can see they overall have a similar fold. You can display these the way you would display the chain tracing of a protein or RNA. This is the chain tracing of an entire chromosome. And in fact, you can do all the chromosomes in the nucleus. And this is work from Ting Wu's lab, with whom we've collaborated. Her lab is also at Harvard Medical Department of Genetics. Now, just to blow up a little piece of one of those chromosomes, a much smaller segment, to show how high resolution, how close to molecular component scale this is. This is a little loop of chromatin chromosome that is a regulatory unit and is closed by this orange and red segments, which you can see have subsegments here. Each of these balls is an independent microscopic analysis. The entire region that causes loop closure is 10 kilobases, and each of these balls is a tiny fraction of that 10 kilobases. So we're getting down into this sort of the 20 nanometer or nucleosome or few nucleosome range. So these are some of the people that have collaborated on this, notably led by Ting Wu, and we participated in this recent paper in Nature Methods led by Boyd Wynn. So that's DNA, this is RNA. Each dot is a single RNA molecule, changing color but not position. And then we can ask the computer to recite the outcome of all those cycles of next-gen sequencing, where the 4 colors correspond to the 4 bases. And now we have a barcode for each one of those RNA molecules. This was Jay Lee and Evan Doherty. Jay at Cold Spring Harbor, Evan helped start ReadCore, now at 10x Genomics. So that's DNA, RNA, finally proteins. To make proteins similar to the previous two, we tag them with a little 9-mer oligonucleotide, either in the IgG, Y-shaped molecule, or a single domain, a nanobody, can be much smaller. In any case, by tagging it with DNA, we can do DNA exchange imaging, which both makes it easy to image, but it also gives us super resolution. So you can see that here we're trying to visualize the synapses that occur throughout the brain, and are the connections, thousands of connections per neuron. These synapses are key to knowing how the connections go, but you can't see them in the normal microscopy. But in this DNA exchange imaging, you can see up to 4 different proteins that tell us the direction of the synapse from pre to post synaptic, and whether it's inhibitory or excitatory. This is because we're using antibodies to image the proteins at super resolution, sort of 20 nanometer resolution. This was work of Yu Wang, who was a graduate student and postdoc in my lab, and also shared with Hong Yin's lab at Harvard and Wyss Institute. Now, finally, we want to be able to not just take a static image of DNA, RNA, protein, but we'd like to be able to do live imaging or timed-based imaging of activity. And this is an example of calcium imaging done by Ahrens et al. And but it's limited. It's limited to a specific species that happens to be transparent and small, the larval zebrafish. It has to be a thin section, and it is limited in resolution, so we can't see the high resolution I've been showing you. So we'd like to fix that. We'd like to be able to use thick sections, multiple sections, and integrate it with the super resolution we had before. And we do that by converting the time axis into a DNA axis. So we use a DNA recording device the same way that you use a flight recorder in an airplane to record lots of data as a function of time, and then store it so that later you can look at it in a fixed specimen. And we showed many years ago that we could get data storage at densities up to hundreds of exabytes. That's 10 to the 18th bytes per gram. And we do this, so Prashant Mali, who was one of the co-inventors with Wuhan Yang and others of CRISPR, collaborated with us, came back in a certain sense, and collaborated with Reza Kolhar to change CRISPR in a way to turn it into a recording device. So the textbook version of CRISPR here is you make the guide RNA, this little gray arrow is synthesis. The guide RNA binds to the nuclease in purple, it cleaves the target, and then everything stops happening because this target is destroyed. It no longer has anything to recognize. We changed that so its target is itself. It targets its own guide RNA. So even though it's over temporarily when it alters the target, edits the target, it keeps editing randomly, producing a wide diversity of signals, which we can image with this in situ imaging shown here in this science paper. So we, Reza put these homing guide RNAs, these guide RNAs that edit themselves throughout the genome in 40 to 60 different locations, so they wouldn't interfere with each other. If one gets deleted, it doesn't hurt an adjacent one. And this just shows the dramatic resilience of living systems to lots of CRISPR, both on target and off target. This is, dozens of randomly chosen guide RNAs that are getting even more random during developmental biology, going from the single cell egg to the adult animal. without serious health consequences. They get to adulthood, and these mice are available freely through an NIH resource, and we can use the information as we can sequence, targeted sequence on those guide RNAs throughout the genome to see how each cell differs from adjacent cells in the anatomy of the animal, and then we can use that to reconstruct what happened as a function of time. And other labs have used our system to not only monitor and record the developmental lineage, but also the physiological changes, for example, hypoxic response. And we think that this is not obviously limited. So in terms of developmental biology, we can make discoveries when we reconstruct the phylogenetic tree. So each cell is more related to its sibling cells than to any other cell in the adult or embryonic body. And this goes all the way back to single cell, and we can construct these trees. And we discovered that the decision during development of forebrain versus hindbrain occurs earlier in development than the right-left decision. And I should emphasize that we think the amount of data that's being stored, we haven't retrieved it all, but we have stored 4 times 10 to the 12th, or 4 trillion bits of information, almost a terabyte of information, in a very compact state. So just 30 nanograms out of the 30-gram mass, so 1/1000th of the mass of the animal, stores this vast amount of information. So I just want to open it up for questions to close the talk by thanking someone. I've already thanked many graduate students and postdocs in the talk, but I want to mention Ting Wu, whose lab does the DNA imaging in collaboration with us, and Ed Boyden, who's done DNA and RNA and protein imaging using expansion microscopy, and Farron Isaacs, who's a terrific collaborator on GP Right, which is about making these mammalian engineered recoding. And in particular, the mammalian recoding team is now Ariona and Yuting were postdoctoral fellows. Now Ariona is heading the effort for the cold-resistant elephants in Boston, and Yuting is in Shenzhen, China, running his own lab, collaborating with us on this kind of genome recoding. And Revive and Restore has also been very helpful in getting our elephant project going. Stephen Casper did amazing software development for the mammalian recoding. So period, full stop. Thank you very much. I will return control to the organizers. Thank you.
Thank you so much, George, for this wonderful and insightful presentation. I'm sure every one of us have enjoyed it. Now we will take some questions. So I open up the floor to question and answers to all the audience. You can either unmute yourselves and ask your question or type it in the chat box, please. This talk is also being streamed live on YouTube, so I will be looking at the chat in the YouTube section also for question and answers. So as people are getting ready to ask questions, I have, as a moderator, I would like to start off asking about, you talked about turning on human cell RNA landscapes and using them for cell therapies. So do you think there would be noise associated with the microbiome that is present in humans, and how would that impact these the final landscapes, and how do you see them interacting?
Yes, that's an excellent question. Both the epigenetics that we describe for altering the cell fate and the microbiomics have been mysterious until recently. I mean, of course, they still are mysterious, but that we've made breakthroughs now that both of them are making it into medical applications. So we don't have complete understanding, but we have enough. And so we have some medical products where we engineer the microbiome. For example, one of my companies, Nella Fit Biomics, takes microbiomes from elite athletes. The epigenetic reprogramming of stem cells into, say, these organoids that rescue demyelinating diseases, there are formally no microbial components of that system. It's all done sterilely. There are endogenous retroviruses, but they're not. But they're human into human, or a pig into human. In the pig case, we eliminated the endogenous retroviruses. So this kind of epigenetics has been separated from the microbiome. They definitely interact, so that your gut organisms will send metabolic molecules into the blood, and then that can affect brain function. For example, the gamma aminobutyric acid is an example of a. So you need to be aware of all these complex system-wide interactions, but those kinds of transplants are typically done in a way that should not affect the microbiome or vice versa.
Thank you. I think one of my colleagues, Dr. Dhanasekaran, has a question. Can we unmute Dhanasekaran so he could ask his question?
He can unmute himself.
Yeah, hello, Professor Church. So it's really a fantastic talk. So my question, I just put in the chat, but the same question I can ask, which is, how do you monitor genome stability in this, modified, when the genomes are heavily edited at multiple locations? So there are a couple of questions. One is, how do you monitor that for many generations to ensure this edits are maintained? Is there reversal of edits and so on and so forth? And I know this animals can be maintained isogenic crosses, but with crossbreeding, will these edits, ultimately go away? And how is that sort of tackled?
Right. So I mean, there's essentially great question. There's two heavily edited animals that I discussed. One is the pigs, where we have 42 edits that are used for transplants, and then in the mice we have 60 edits that keep changing. The first one is intended to be stable. They're intended to be homozygous, so that when we do the inbred strain, they breed true, and they breed just like any other strain of pigs. In fact, we've done it in multiple background strains, and it is stable. And the way we establish genome stability is by whole genome sequencing. And we now have multiple generations and dozens of healthy pigs that are being used for organ transplants in these preclinical trials. So that, I think, is fairly well established stability. For the mouse, that is intentionally unstable, and we monitor the instability at each of the guide RNAs by targeted sequencing throughout the lifetime, and that's the basis of the method. They are stable between generations because we keep the CRISPR separate from the guide RNAs, so they're in separate strains and they're only crossed in when we want to do an experiment. It nevertheless is challenging to maintain the strain because they're not homozygous, like the pig is homozygous. In this case, they are not, and that means we have to check them at each generation by standard breeding methods, where you'll take PCR analysis from skin from the animal. But this is not done by us. This is done by an NIH resource where they are. We can also freeze them, so you can freeze the embryos, so we can always go back to them if it starts to drift.
Yeah, thanks. That's amazing that we can actually follow these animals by sequencing for many generations. So the last question I have is going back to the beginning of your talk. You talked about how we can knock out this HLA antigens to get tissues which can be universal donors, which don't have the rejection problem. But what about the native functions of HLA antigens? They have important functions in terms of immune response and just the tissue modeling and et cetera, right? So
right.
how are those native functions compensated in this in this modified tissues which are donated?
Right. So what we use is inspired by an observation that in humans and many mammals, when after pregnancy, the mother's blood has circulating fetal cells in it. So these fetal cells have most of the immune functionality, and they survive for many years, sometimes decades, in the maternal system without being rejected by the mother. They should be rejected because they have the father's antigens in them, but they're not. And that's because they have, instead of the normal class 1 and class 2 MHC, they have HLA-E. HLA-E has some of the functions of class 1 and 2, but is not rejected. So that's one of the strategies that we're using to answer your question.
Yeah, sure. Thanks a lot. Yeah.
The next question is by another colleague of mine, Dr. Premnath. Can someone unmute Dr. Premnath so he can ask his question directly?
Hello, can you hear me?
Yes.
Yes. Professor Church, I wanted to request you to comment a little bit on how you see the future of gene therapy using some of these base and gene editing tools evolve in the coming years. The last one we heard about was, I heard about was ATTR, but how do you see it evolving over a period of time? What are the diseases that we can expect to see being resolved through gene therapy in the coming years, especially in the next, say, 5 to 10 years?
So 10 years is about the amount of time it normally takes to go through a challenging clinical trial. The very shortest clinical trials are about 1 year now, say for vaccines. So 1 to 10 years is how long it takes. But many things are happening in parallel. So that those 10 years will see a revolution. And gene therapies were initially aimed at rare diseases, but I think now we're seeing them focused on common diseases. For example, the vaccines, unfortunately, very common disease, but also for aging. So essentially all diseases are impacted by aging in one way or another. So a gene therapy that's focused on that could be very impactful for a variety of diseases, some of which are actually fairly early onset, adult onset, but early. And so we have one such drug, which is combination gene therapy of 3 different genes that has been through mouse studies and clinical trials in dogs for a veterinary application for dogs that die prematurely of things like mitral valve disease. It's aimed so far at about 7 different age-related diseases with one drug cocktail. And as soon as the dog studies are complete, we will begin human clinical trials. So that's an example of where, now that's gene therapy, that's not CRISPR, that's addition of 3 genes that you already have, or addition of regulatory factors that regulate those 3 genes. So there's no immune rejection of those, and it could be a permanent change. So I think that's example of what could be happening in the near future. They seem to be, as a category, they can be very safe and very low cost, which worried me when we first started on this, but it is less concerning now because of the tremendous success of the 3 vaccines, AstraZeneca, Pfizer, and Moderna.
Sure. Thank you.
Yes. Can you hear me, Dr. Church?
Yes.
So I was wondering, proteins, barcodes, and are very high resolution, and they're very valuable in assessing the interactions between cells, including the synapses that you talked about. So can you comment on some of the futuristic directions that one can take with this technology to understand neural connectivity, and also, what are the ramifications for mapping the human brain and treating Alzheimer's disease or any other neurodegenerative disease?
Right. So for many disease diagnoses, those can be done on very small samples, but at very high resolution. So I think that's a good combination. If you want to understand the entire brain, that's much more challenging. But although it's less of an acute problem, it's not so much of an immediate medical problem, so much as growing our background knowledge, our basic knowledge, and that is we're trying to accelerate research on the speed at which we can acquire these data. There's a new generation of microscopes that are probably 100 times faster. And I think that combined with parallelism should allow us to get a complete connectome, at least of small rodent brains. And then we can do genetic engineering of those brains to test hypotheses. So we can do serial sections and get not only the DNA, RNA, and protein, either targeted or random shotgun, but we can also get these time series data that I was talking about, where you can get developmental lineage and physiological changes over time. So I think if you have that collection done at high speed and high resolution, will be magical in its impact on neuroscience. And that was why we helped start the BRAIN Initiative back in 2011. And that's, in the United States, that's been a very impactful technology development project.
So there's a question from Shikha. Shikha, can we have her unmuted so she can ask her question?
Evening, everyone. Nice talk, sir. And my question is, has there been any attempt on reversing the effect of teratogens, or do you see it as one possibility in the near future? And as you also mentioned in your talk, that real-time monitoring of the DNA, RNA, and proteins is pretty much possible. So do you see it as a possibility to stop repercussions of the teratogens then and there only?
Okay, so the first question about teratogens, some teratogenic effects are hard to reverse without, say, transplants. But the things I mentioned about transplants would be one way of doing that. So some, children are born with, missing limbs or disorganized organs. So that's organ transplant probably. I think preventing the teratogens would be even more impactful. So that's the first question. The second one, we didn't really demonstrate real-time monitoring. We showed time monitoring, but after a fixed point. So real-time monitoring we are working on, and I think that would be very important to get a closed feedback loop where we can make a change, see the effect of that change, and adjust it with machine learning or something like that. The closest we had to real-time monitoring was the Nanopore, which is sort of maybe a 20-minute latency delay, but we hope to get much faster than that. There are real-time imaging methods, but as I mentioned, they have a very short working distance, less than a millimeter, and a low resolution, sort of on the order of half a micron. So, but there are things that we could do with that. But anyway, that closed loop real-time monitoring, I think you're right, is an important part of our research and medical practice.
So, Anu, can I ask one more question? Then a second.
Sure. Please go ahead.
Professor Church, just one question regarding the sequencing technology itself. We have seen, tremendous changes in the past decade or so, but what do you think is the next 10 years in sequencing? Are there a more faster and more robust technologies in the pipeline, which makes it more like something like PCR sequencing in every lab, like a PCR technique in every lab? Will sequencing be commonplace? I think it should become like that, but are there new technologies in the pipeline going towards that direction?
Yeah, there are. I think the most promising ones are two kinds. This is for portable, easily accessible, inexpensive sequencing. In addition to the microscopy that I described, let's talk about the portable, inexpensive sequencing. There are two types. One is the nanopore. Two types of nanopores. One where the single-strand DNA goes through the pore. One where a polymerase pushes it past the pore, where the triphosphate is detected. And then the third one is a molecular transistor that's going to be announced by Roswell. Well, it's already been partially announced by Roswell. So molecular transistors and nanopores. These are the chips. Part of them are tiny. They're smaller than your fingernail, and usually the device is bigger so far. But they will probably be integrated into cell phones, so it'll be totally portable and ubiquitous. They can be either in fixed positions in high traffic areas, or they could be real-time monitors on people's clothing that they carry around with them to monitor their food, their air, and their water. So I think that will be a revolution in monitoring. It would protect us from zoonotic diseases, including some of the horrible ones we've had recently, as well as emerging resistant variants, resistant to vaccines, resistant to drugs. If we have literally millions to billions of these monitors at low cost, constantly monitoring everywhere that there are people, and even places where there are not people, that would greatly help our preventative medicine, our global preventative medicine. We sometimes call this a bioweather map, where we have both very fine structure, local measurements, as well as a global context and a global discussion about emerging diseases. I think this is extremely important.
Yeah, that's very nice. Thanks, Eddie.
So Dr. Church, based on your experience, how much do you think we can reduce off-target effects during human genome editing? Can we use these self-coding homing guides that you mentioned for that? And also in the same context, how accurately can one predict the phenotypic effects of these genome edits?
Right, so off targets for the classic, the original CRISPR were not so bad. So the original CRISPR that we published in 2013 was Streptococcus pyogenes Cas9, and with a properly designed guide RNA, a properly tested guide RNA, you could get lower levels of off-target than the spontaneous mutation rate. So if you're going to, you don't need to worry about it any more than you worry about spontaneous mutation. It may be 1 in 100,000 cells. The place where it starts to become problematic again is when you're trying to get these base editors, these more precise base editors, like especially the cytosine base editor. That has a lot of off-target, and you know, some labs are working on that. An alternative to the base editors and an alternative to CRISPR in general is our recombinases, where you pop in a piece of DNA where you have to check both sides have to match the genome, and the recombinase has to match. And so all that checking results in much more faithful, and the replacement, you replace it with a piece of DNA that you've sequenced in advance, so you know that it's the right sequence. So I think that's a very promising direction. One of my companies called Tessera is pursuing that, in particular with recombinases that are derived from the human genome, so they're not even foreign material. CRISPR is a foreign material from a bacteria. These recombinases are from the human genome, so they're native.
Nice. We have one more question by Dr. Premnath. Premnath, would you?
Yes. I wanted to ask you about something which is not strictly within the scope of what you spoke today, but I hope you can address it. One question relates to some of your efforts in the clean energy space and startups in that space as well. Can you tell us a little bit about those efforts, including LS9, for example? And the second one, a quick question is, as a researcher, how do you go about creating some of those startups? Because I think you have been involved in more than 20 startups now, it appears. So, and what has been the motivation driving the creation of those startups?
So clean energy or biofuels is a very challenging field because it's very inexpensive to just pull fuels out of the ground. It's not responsible, but it is inexpensive. And so that's what we're competing with. LS9 and another one of my startups, Joule, did very nice science. They discovered an enzyme that produces alkanes, meaning hydrocarbons, like gasoline and diesel fuel. And they did that by a decarbonylation reaction. So they each discovered this pathway. And they were merged as companies with companies that produce a lot of carbon dioxide, so that they work better if there's a high concentration. So in the atmosphere, it's only 0.05%, but the factories sometimes are producing at 30% carbon dioxide. So that's a good pairing, but it's still not quite good enough to impact the world yet, anyway. So what we're doing now is working on carbon sequestration. So rather than working on the fuel side, working on pulling carbon dioxide out of the air. And that's better anyway, because even a biofuel, you just get one extra use. You take the carbon dioxide,
yep.
Turn it into fuel, you burn it, it turns into carbon dioxide again. So there's a net zero. What we need is net negative. We need to remove the carbon dioxide permanently out of the air. And that's what our carbon sequestration is all about. For example, one of them that got a lot of press is the elephant, but it's not the only thing that we're doing for carbon sequestration. The Arctic is a particularly good place to do that. Surprisingly, some people are surprised because they don't think of the Arctic as being a heavy photosynthesis, but it actually has more fixed carbon already than the rainforests, and that's because each year it's sequestered in a frozen layer. That sequestration is important. Now, to your second question was, how do we start these things? I've co-founded 38 companies now. Most of the most recent ones were done by postdoctoral fellows who had made an invention in the lab and then wanted to do a startup. And we've been very fortunate. The last few years have been good for investors, and so the investors like some of the things that we've done in the past. And Boston is a particularly good environment for such investments. There's no magic formula, but it mainly is hard work and having made some invention prior to forming the company. You want to have it de-risked as much as possible in an academic setting before you start the company. I hope that's helpful.
Sure, thank you.
I'm sure we can go on for many hours or the whole day asking George questions, but it's almost hitting our hard stop mark of 6:20 p.m. IST. So I'd like to thank George for answering all the questions. It was a great pleasure to have you with us virtually. I wish you were here with us personally so I could get my book signed. I would recommend all the audience to purchase a copy of this book and read it. It's called Regenesis, and it's a very provocative attempt at describing how synthetic biology can be used to reinvent ourselves. So thank you so much, George, for your time and for being with us. Before we conclude, I would like to thank our whole event management team for helping with all the logistics, the meeting links, the IT arrangements, you name it. So this concludes our 76th R.A. Mashelkar Endowment Lecture. Thank you all for attending it and being a part of this wonderful event. I hope you have learned and enjoyed this fascinating presentation on the analysis, synthesis, and imaging of very large molecules. I was really excited by the title, George, and it was really brilliant. Thank you so much, and I hope we can interact with you further.
Thank you very much. It's been wonderful to have this conversation, and hopefully many more in the future.
Thank you.
Thank you.
Bye-bye.
Bye.
Thank you, George. Thank you.
Bye.